You don't want to ask questions?
Should I go?
Yeah. Yep, whenever you're ready.
So, hello everyone and welcome to my presentation on, well, the title is Are You Scared of Water? So, hydrophobicity, hydrophilicity, and amphiphobicity. So, before moving into all of the information that I'm going to present to you, it's important to understand the meaning of these three terms. So, hydrophilicity is the property of any molecule in which it's ionic and therefore it's attracted to water and it can dissolve very, very well in water. And ionic obviously means polar, so it has a charge. For hydrophobic substances, they're not necessarily charged and they're nonpolar, which means they interact better with neutral or other non-ionic or nonpolar substances than with water. For amphiphilic substances, they hold the properties of both nonpolar and polar substances and are usually have areas on the molecules themselves that are polar and are nonpolar, and we'll see that later in the presentation. So just as history about hydrophobicity and philicity and amphiphobicity, hydrophilicity and amphiphobicity have been used very often and have been discovered in our bodies and discovered everywhere else. But hydrophobicity was researched very, very deeply. The first person to actually research the concept of hydrophobicity was Robert Wenzel, and he outlined the basis on hydrophobic substances. And basically, what Wenzel did, he knew what hydrophobicity was and what it caused, and he had a theory. It's a little hard to see here, but this was Wenzel's idea, where the contact angle is a little bit more than a drop of water being on a non-hydrophilic substance, so a non-hydrophobic substance. So his contact angle was slightly more than 90 degrees, and the water, since hydrophobic substances tend to have ridges that come off of the material themselves, in his idea, the water would fill in the spaces between those ridges. And after him came Casey and Baxter. These two were scientists that believed that water was more slippery than Robert Wenzel's idea was. Their contact angle was very larger than Wenzel's contact angle, and the water rested on top of the ridges instead of going in between the ridges. And just as many breakthroughs that have been discovered, one more breakthrough was the discovery of the super hydrophobicity of the lotus leaf, and this was done by Wilhelm Bartlett and with his partner, and he discovered how this lotus has a hydrophobic coating, natural hydrophobic coating. So now, moving on to the importance, usage, and applications of these hydrophilic and amphiphilic substances. So basically, I'll answer the questions like where can we use these, where can they be found, where are they naturally occurring, and basically what is the application in the real world. So for hydrophobic substances, we have applicable coatings. And these are basically coatings that you put on other substances to make them hydrophobic. One such example is Neverwet, and that's a company that produces sprays that you can spray on many different substances to make them super hydrophobic, just like a lotus leaf. Apparel, obviously, and that's like with our coats. A lot of coats these days are hydrophobic to keep you as dry as possible whenever in any kind of extreme weather situation. Construction is a very important one, especially with you can use apparel during construction too. Like if you're in mid construction and you don't want anything to get ruined or not necessarily ruined or anything to get wet, anything important to get wet, then you can apply a hydrophobic coating to it and just while the construction is going on and make sure everything is safe. And also workers' boots during construction have hydrophobic coatings also because construction involves cutting and going into many unfavorable situations. So the hydrophobic coating on the boots can protect them from any liquids or water that they may get on their shoes. For hydrophilicity, it has two very important medical uses. Well, one is the medical use and one is a medical application that we can see. For the medical uses, hydrophilicity is used in catheters. And basically what this is a catheter is basically a tube that's inserted into a person, whether it be through the anus or whether it be through any other, the nose. Sometimes they have a nasogastric tube, and that any kind of catheter is that can be used to draw blood, insert chemicals, maybe for IV. And basically, a hydrophilic catheter will help the movement of water and other substances through the catheter more efficiently than a catheter that doesn't have this coating. So it allows for the smooth transport of liquids. For diseases, hydrophilicity is a very important concept in our own body, because many amino acids, which are proteins that help manufacture, which are monomers of proteins which help manufacture many proteins that are essential for life. They have important hydrophilic and hydrophobic properties. So one such important disease is sickle cell and sickle cell anemia. This is a disease where glutamic acid, which is one of the 20 amino acids that are required for life, is changed to valine. And glutamic acid is strongly hydrophilic. And when it changes to valine, valine is a very hydrophobic molecule, and it's a point mutation in one of the codons in our genome. And when this mutation is caused, it causes the hydrophobic substance of valine to form and attach instead of glutamic acid, and this causes the sickling of our cells. And because the hemoglobin on our red blood cells doesn't properly function, and the sickling of our cells can cause to blocking of our arteries, and thus not proper transport of blood to many parts of your body, and this can cause many problems like fatigue and maybe even death of a part of your body or because of lack of oxygen. And finally, amphiphillicity has a very important application in our own body and many other places. In our own body, we have millions of cells that make up all parts of our body, and each individual cell needs nutrients, and without it the cell would die. The nutrients that are passed into the cell are passed through many different processes like facilitated diffusion, active transport, but it needs a membrane to actually pass through. And this membrane is made up of a lipid bilayer that made of components called phospholipids that I've drawn here. And each of these phospholipids it represents an amphiphilic molecule because they're composed of two parts, which is a hydrophilic head and a hydrophobic tail. These two separate characteristics of the parts of these phospholipids presents a very important structure, because it allows passage of some molecules into the cell and some molecules out, and it forms a semi-permeable membrane is what it's called, when it allows to only certain molecules or certain substances to pass into the cell. And this semi-permeable membrane is what allows our cells to live and grow. And so that's just background on the topic of hydrophobicity, philicity, and epiphilicity. Now, moving on to the procedure, the actual experiment that we carried out to relate to this idea. So in our experiment, we had 4 parts. In the first part, the procedure was that we mixed 2 vials. One vial had ethanol and water, and 1 ml of each. And we mixed those two. We put those in a vial and mixed those two together, and we let that sit for a while. Initially, the observation was that it was a very foggy substance, but we let it sit for a while. And meanwhile, while it was sitting, we took another vial. We put 1 ml of water and 1 ml of vegetable oil in that vial. We mixed it up together, and almost immediately we saw the obvious results that would happen. And those will be shown in like a little bit. And then. So we let them sit for a while, for about 2 minutes, and in the ethanol vial we saw that the liquid went from a foggy kind of substance to a very clear substance, and this proved that ethanol was a very hydrophilic substance because of its polar properties, because of a hydroxyl group that's attached onto the ethanol itself. In this diagram, I've circled the hydroxyl group, and that's what gives it its hydrophilic properties. For the vegetable oil is a lipid, and Yeah, and it's strongly nonpolar. So when we mix the two substances and let them sit for a while, almost immediately the layers begin to form because they do not mix properly. And this is also known as self-assembly, which this is also part of self-assembly, but when the two substances separate, they're assembling themselves together, and the barrier that's formed separates the two substances completely, and this is called the property of self-assembly. So after those results, we figured out which one was nonpolar, which one was hydrophobic, which one was polar and hydrophilic. After that, we tested out some actual substances with water. So we used plastic and we used glass. So normally, even in the lab, we thought, all three of us thought that glass would be the more hydrophobic substance. So when we put a drop of water on glass, that it would stay as one mass, and because we usually see teardrops on glass and they look like little buds, so we thought that maybe that would be a more hydrophobic substance and the plastic would be hydrophilic. But that was proven wrong, and we saw that when we placed a drop of water on a glass slide, the drop of water spread out completely, and that's because of the electromagnetic properties of glass, and because it's negative, it's a negative substance, that water spreads out more easily, and it's more hydrophilic, and plastic was more hydrophobic because because water didn't interact properly well with it as well as glass. So I just printed out there's a diagram showing what the contact angle with plastic would be larger than with glass because the glass was a more hydrophilic substance. And in part 2 of our experiment, we made 3 separate solutions. We used 2 of the solutions, and in these we used the ethanol vial and we used the vegetable oil vial. And the third solution we made was with milk and water. So we have ethanol and water, we have vegetable oil and water, and we have milk and water. And in this we wanted to observe the formation of these structures called micelles and a colloidal suspension. So basically what that is when two substances are within or are immersed within each other, they can exist as they can exist within each other even though they retain their properties even though they may be different from each other. For example, milk is like a solid within a solid. Smog would be like a solid within a gas, and there's many other forms of these kinds of colloidal suspensions. Basically, we tried to see what kind of solutions were formed when we shined a laser into these vials. So in the first vial with ethanol, we took the vial with ethanol and shined a laser directly through it into the wall, and it showed minimal scattering, and it showed you could see the dot coming of the laser through the vial on the wall, and this was important because it showed that ethanol was a rather homogeneous or uniform substance, and it didn't allow for much scattering because there wasn't the formation of it wasn't a large colloidal suspension. When we did the milk, though, the light completely didn't pass through at all, and this is because of the congregation of the micelle that formed within the milk. I did some research at home, and the name of the micelles that are formed within the milk are caseins, and here's a diagram right here. It's pretty small, but the caseins are basically the micelles that form within the milk, and they are formed with calcium and phosphate bonds, and those are the solids that are put within the milk that don't allow for the laser to pass through. The last substance we tested was oil and water, and this was a very, very quick procedure because while we mixed it, the layers already began separating, so we had to shine the laser immediately. And we noticed that it glowed just like the milk, and this is because of the substance of the micelles that were forming intermediate between the layers separating. And then part 3 was really quick. That was in preparation for part octadecane. We just applied a substance called octadecane thiol on a silver film. And what octadecane thiol is well, any thiol is a substance that has a sulfur group, and it has specific properties and usually is attracted to other metals. And it was seen in this in this experiment. We'll get to that in part 4. So we just, step 3 was just the application of the octadecane thiol. Part 4 we used. Well, the first part was we had some fun with this. We used a piece of cardboard with a never wet or super hydrophobic coating just to see how a super hydrophobic coating works. And so we used a pipette with water and then tried to keep the water on the super hydrophobic surface. And it took a while, but it just showed how hydrophobic these substances can be made. And then we did another super hydrophobic experiment, or a super hydrophobic experiment with a glass slide, in which the edges of the slide were sprayed with this hydrophobic substance, and the middle wasn't, and the middle was just glass. So as we saw before, glass is hydrophilic, and hydrophobic substances are not hydrophilic. So when we put the water in the center of the glass slide, it stayed as one mass in the center of the glass slide, didn't pass outside as it would normally be expected, because it was attracted to the glass, and it was not attracted to the hydrophobic edges. And then we moved on to the silver film, which we used for the application of the octadecane thiol. And in this one, what we did was we used the cardboard, we used a normal silver film, and we used the octadecane thiol silver film to see what the properties of these substances are. So just working from the bottom up, obviously the cardboard was a very hydrophobic substance because of the spray that the hydrophobic spray that had been put on the cardboard. The octadecane thiol, well, we should move to the bare AG film, silver film first. So this film, the water spread out very much, and it formed a pretty low contact angle, and this means that the silver itself was attracted to the water much more than any hydrophobic substance would. And when we applied the octadecane thiol, the contact angle suddenly of the water became high. And what this means is the water began to clump up and form these balls because it suddenly became hydrophobic. And this is seen in this diagram right here. This is what an octadecane thiol molecule would look like. The bottom says Ag, so silver, that's like a silver film. And these are the octadecane thiol molecules. The heads are attracted to the silver, and these tails, which are hydrophobic, they form a barrier above. So they essentially work to form this hydrophobicity on the silver, and that wouldn't be seen on a normal bare silver film. So that was our experiment and all the observations that we had. This is just a summary of the observations that we did. So as for our results, we observed that ethanol was a very polar molecule and mixed well with water. Vegetable oil was nonpolar and thus did not mix well with water, form two distinct layers. Plastic was more plastic was more hydrophobic than glass, and it had the clumping of water more than glass did. Ethanol minimally scattered light, which means that when we shined a light through it, it was a homogeneous mixture, so it allowed the passage of the laser through. Milk and oil, they glowed in water, and this is because of the formation of these micelles and these colloidal suspensions. Super hydrophobicity, we demonstrated that with the cardboard and kind of demonstrated the lotus leaf. Oh, there's one more thing I forgot about super hydrophobicity. It allows for this technique called self-cleaning, and what this is when a material is super hydrophobic, water can pass over it very quickly, and what this means is it picks up any substances on that coating, but retains the property of the hydrophobicity, so it can allow for quick cleaning of these substances. So that was just back to the superhydrophobicity. And back to the results, water was pooling in the center of that glass slide because it was glass is hydrophilic, hydrophobic substances are not hydrophilic. And water reacted differently on silver slides with octadecane thiol, and on superhydrophobic substances. So as for my conclusion, I said that from our results we can learn many different substances have many different properties and thus react in separate and different ways in water. And the improvement that I thought we could make in this experiment and make it better is that we could test more substances just to see the wide range of properties that every substance possesses. And I said for the future, we should keep in mind to always work quickly, but always understand concepts as we're moving through labs. And these are just some other important figures that I thought I should put on here. So this was I noted to before with the milk, and this is the casein micelle that the milk forms. This is the octadecane thiol layer that forms on the silver film. We worked with the cardboard, but I couldn't find a cardboard picture, so I found a newspaper that was also sprayed with this hydrophobic substance, and just to see how this one got wet and this one the water stayed on. This is just a quick briefing of contact angles, where if there's a hydrophobic substance, it's going to have a large contact angle because the water is not spreading out as much, and thus it forms these clumps of water that don't spread out as much as on a hydrophilic substance. This is just water reacting on a lotus leaf. Well, not reacting. Water. Lotus leaf's hydrophobicity in action. This is this is oil and water separate because of their nonpolar and polar qualities. This is what we observed when the milk with milk in the vial that we had, because the vial glowed and it didn't allow for passage of the laser through. This is just a summary of all the different kinds of structures that can be formed with the amphiphilic molecules. But the two most important ones are C, which is, if you can't see that, we probably can't. The blue is the tails, the hydrophobic tails, and the green is the hydrophilic heads. So the B is showing just my cell forming because the tails are inwards and the heads are outwards. And this is a lipid bilayer because it's a straight structure with the tails inwards, heads outwards. And this is just another diagram with these molecules in their formations. And so that's my presentation. Thank you for listening.
Thank you, Megh. That was a very nice presentation. I have quite a few questions that I plan on asking, but does anyone have... Yeah, you can get started.
Okay, so chemically, what makes a substance hydrophobic and hydrophilic?
Hydrophobic and hydrophilic, it's the fact that they're either polar or non-polar. Polar substances, they technically are they technically have more attraction. Just to take an example, polar substance is water. The oxygen has a higher pull on the electrons that are shared with the hydrogens, and this causes the water to be more electronegative and the hydrogens to be positive. And this kind of polar substance will cause hydrophilicity. Hydrophilicity, and this is seen with the ethanol. Sorry, as I showed you. And then for hydrophobic, it's not polar, and it's usually very stable and neutral.
Do you have an example of a nonpolar molecule?
Non-polar molecule, any kind of oil is a non-polar molecule. Most kinds of oil, and yeah.
Actually, I have a follow-up question to that also. So, what's the difference between a chemically hydrophobic surface and a physically hydrophobic one?
Like plastic and oil.
Okay, I see. So a chemically hydrophobic substance will not contains properties of non-polarity, and that's non-polar and doesn't react well with any kind of polar substances. Physically hydrophobic can be with the use of nanostructures that I should draw this. So just to take the example of the lotus leaf itself, that's a physically hydrophobic structure, while oil isn't chemically. So oil just is chemically hydrophobic because it's like because it's nonpolar, but a lotus leaf is physically hydrophobic because it contains these nanostructures. Well, this is a microstructure well, it contains millions and millions of these everywhere, and this nanostructure, microstructure, has many nanostructures that are attached to it, and this forms a very rigid kind of structure on the lotus leaf itself. So when a water molecule comes along, it's going to just rest on top, and it's going to slide off of the leaf itself. So physically hydrophobic is just, it contains these rigid kind of structures that don't allow water to stay on very well, and chemically hydrophobic is usually just not polar.
And actually, just as a follow-up question, because I think he started that with like an example of plastic and glass. So is plastic a chemically hydrophobic or physically hydrophobic surface?
Plastic is a physically hydrophobic.
I think I would actually say it should be chemically.
Oh, okay.
It's mostly just made from organic polymers, materials.
Physically hydrophobic, are they crystals? Is that why? Because they're densely packed? I mean, you did talk a bit about the silver, which is from the drawing, a crystalline substance, which I didn't realize silver was crystalline. Because of that shape, is that what causes it to be physically?
It is because of the shape, and it's because as I showed you here, and I showed and you saw here, the shape of the microstructures itself, and the structures coming off of them, they form this rigid kind of material that allow the water to pass off of. So it is because of the shape that the shape is what determines the hydrophobicity of the substance.
Now, I didn't understand the connection between the experiments you did with the laser and whether or not something was, it seemed like that had nothing, I didn't get the connection between that and whether or not something was hydrophobic.
I see. So basically what the connection is that these micelles that are forming, they're forming because of their hydrophobic properties, and
So the hydrophobic suspension is what you're saying.
Colloidal, well, it's called a colloidal suspension, but basically the groups that are forming, which are the micelle, is because of their shared properties and of hydrophobicity, and then they form together, and when you shine a laser through that, that's the suspension that forms. So those are the molecules that it's bouncing off of.
Yeah, there's the degree to which something is hydrophobic or hydrophilic. It's not like a yes or a no. Is there a measure to that you have like some, right? And what is that?
So if a substance is considered hydrophobic, it's usually greater than about 150 degrees. So super hydrophobic would be well.
Maybe can you like draw out
the degree
like you have this diagram on your poster, but
right.
can you just redraw it on the board?
Okay, yeah.
And show exactly where the thing is.
Okay, yeah. So if we have a hydrophobic, should I erase this? Or?
Yeah, you can erase it.
So if we have a hydrophobic substance, well, let's say like it was like a dividing line, and this is philic, and this is phobic. So in a philic substance, so basically if we put a straight edge next to the droplet of water that going up this way, this angle of contact, which is called the contact angle, would be about less than, usually if it's very, very hydrophilic, it could be less than 20 degrees, and it could be in between about 20 to nearing 90, but a little bit less than 90. Usually phobic substances that form are, the angle is going outwards that way, and so this angle is very large.
So this is beading, is what we're seeing.
Hmm?
On the phobic, it's
beading. Yeah, exactly.
Got it.
You see beads every like you said, beading. So it's very large. Some substances, as I said before, are about like 150 degrees of this contact angle. Some maybe more if they're super hydrophobic, and so basically the extent to which a substance is hydrophilic or hydrophobic can be measured with these contact angles.
Now, how do you actually measure that contact angle? Did you do that in the lab, and like what?
No, we didn't.
No.
We didn't do that.
How does that get measured?
We approximated it in the lab just by looking at it directly, but
Yeah.
they actually have some contact angle meters where you just very carefully lower a drop of water onto a surface, and you look through like a microscope projecting onto a screen, and there's in the eyepiece actually has a little ruler that you can adjust the angle, and by placing it very carefully where he has the lines drawn, so tangent to where the bead is or the spreading is, you actually measure the contact angle. It's actually just called a contact angle meter. Very nice. So going back to the laser question, how does did we use the principles learned in this lab in any of the later labs? Like, did we revisit any of these principles in any of the later labs?
We revisit the. When making we did revisit the principle of of putting like coating substances basically when we made the layers on other other nanostructures on layers, and we also revisited aggregation of molecules it wasn't the same kind of aggregation, but like when these micelles form these amphiphilic molecules are aggregating, we saw that with surfactants and in later labs. It wasn't the same concept, but we revisited the concept of of molecules coming together basically, and so that's yeah.
So what do we call that concept of molecules coming together? It was the principle of self-assembly.
Oh yeah, okay, yeah.
Just slipped. Yeah.
It's okay.
Now, you mentioned several medical applications
Mhm.
for this. Is there research going on in those areas, and what do you think is the most important place for us to do research in that?
Okay, so there is a ton of research going on with this topic with this topic, and I believe that the most important topic, the most important area is obviously our cells, and it's usually has to do with all of our cells, but a lot of research is going on with many diseases that cause crenation, increasing in size, and many diseases of our cells, but I believe that the most important application is the larger scale work we do with these substances. Like I said, the catheters, because if we find a way to transport large amounts of good chemicals, and not good chemicals, but large amounts of nutrients into our body properly, then we can focus on the smaller stuff more easily. So I believe that it's important that the research that we do on making substances materials like catheters and other materials properly hydrophobic or hydrophilic will help us have large scale increases in delivery of proper medical treatment.
Why did you pick this topic?
Well, I picked this topic because I honestly want to go and do something in the medical field, and I enjoy studying medicine a lot, and so basically this topic related more than any other topic to the medical field. So, and I decided to use that. So that's why I chose this topic.
So would you be interested in continuing your research in this topic at this time? And if so, which direction? Because there's a lot of, it's still a broad topic.
Mm-hmm.
There's a lot of specialties within that. And is that something that you think you could do in the near term, or?
I definitely could, but it'd probably be over a long span of time. It'd be over a long time, because it takes I probably won't have access to everything that I need for research right now. But if I had everything that I could use, then I probably could. I'd have to start somewhere.
What would you need?
I definitely I could use microscopes and obviously, but that would be too just general information. But I could use cells and
It's hard to define.
Yeah, exactly.
That's
Right.
the first step, is to actually define
Mhm.
project.
Mhm.
Yeah, I think the first step would be to look into like the literature and see what's currently going on in terms of research with these subdivisions of the topic, and then once you see what types of experiments are currently ongoing and where there's need for future research, that's when you would have to see what kind of things that you would need.
The most important, though, I believe, for this topic is the manufacturing of our proteins in our body. So if I was to do research, I'd probably do it on the proteins, because in our body, amino acids, which are make up proteins, have a vast amount of properties that lead to them being hydrophobic, hydrophilic, or any kind of, or either of those two. And these proteins make up vast amounts of important substances in our body. So if we could tackle any mutations or problems that happen in those proteins themselves, you could save a lot of you could improve the quality of all of the proteins and or important nutrients manufactured in your body. So that could be one of the most important, and that's what is one of the most important research areas in this topic.
What background did you have in this topic before you took this class?
Before I took this class, it wasn't that much. The most I had learned was in middle school, and that was just like basic ideas, like hydrophobicity being repelling water, hydrophilicity being attracted to water, and basically ionic and nonpolar. It wasn't as in depth as I had an understanding after taking this course.
You didn't have any chemistry knowledge either before this class?
No, not before this class.
Okay, you're looking at me, so that's my question. So, what makes the amphiphilic substances, like, you have like the polar head and the non-polar tail, but like what exactly happens to create such a substance, like chemically?
Oh, okay, I see, yeah. So,
how is like the bonding and stuff?
So basically, the hydrophilic heads, they're basically a phosphate group, and if I could draw it, it's not exactly represented like it's usually represented like this, but in the molecular structure of it, there's a phosphate group right here, and then it's attached to two fatty acid molecules next to it. So this is technically what a phospholipid should look like, but we just simplify it by using it like that. So it's a phosphate group attached to two fatty acids, which is a.
So does it naturally occur, or is it like produced by the cell, or like if a scientist would say want to use it, how would they produce amphiphilic structure?
So
could they like do something like with using polar heads and hydrophobic tails and combine them?
I'm not sure if they could like manufacture them by themselves, but I know that the cell has many processes, like the smooth endoprensile reticulum synthesizes lipids and helps in the formation of these substances. The proteins are manufactured that signal that aid in the creation of these lipid bilayers. So the cell has naturally occurring mechanisms that aid in the manufacturing of these bilayers, but I'm not exactly sure if scientists could manufacture them on their own.
Anybody? Do you know?
I'm sure that they can make these types of things because, in fact, we can purchase something like octodecane thiol, which is technically amphiphilic, because it has the thiol head group and that has the long 18 carbon chain tail. I don't know exactly how they make it though. They probably have to functionalize they probably take a long carbon chain and functionalize one of the ends with the thiol group. I don't know the exact chemical reaction that they would do to functionalize it though. But definitely I'd say they can make it in the lab as well. They could even make the triglycerides probably in the lab.
How is this class different than the science classes you take in school?
Well, this class is definitely more hands-on because every week we come in, we have a new lab. Science classes is basically a lot of theory, and then we do one lab based on all of the theory that we have. In this class, we get to learn the theory, and it's in depth, and it's explained very well because of the handouts and because of, Chris teaches really well. And then after that we apply it. We directly apply it into our labs. We don't waste any time for like the information to like be lost, and it's very fast, and it's very hands-on. So that's how it's different from any school science course that I take.
Thank you. I was very impressed, very impressed. Some of it was over my head, but you did an excellent job. I mean, the research that you did was phenomenal. I mean, certainly not something that I would expect from middle school or high school at all.
I definitely agree.
Very, very, very impressed.
Very nice work.
Thank you.
Thank you.
Take another round of applause.
Good job.